Hi, thanks for listening. I'm going to talk about TensorPhylo, a program that Xavier Meyer and I wrote for state-dependent birth-death models. So a fundamental question in evolutionary biology is how and why rates of diversification vary across the tree of life. Wayne Madison and colleagues in 2007 introduced what's called the BISSI model. This is the first sort of formal phylogenetic method for asking questions of this type. And since then, there's been a proliferation of methods for addressing questions about how particular character states affect diversification rates. So for example, we might want to know whether or not selfing lineages in plants are an evolutionary dead end, or whether lineages in different geographic areas diversify at different rates, or whether or not chromosome changes are associated with speciation events. These methods are also really useful in epidemiology, where they can be used to understand whether different geographic areas confer different rates of spread. So these are really popular methods that have been implemented in many programs, but the implementations have often offered non-overlapping functionality. So some features are available in some, but not others. There are also practical limits on the size of data sets that can be analyzed, and particularly there's a limit on the number of character states that you can analyze. There are many reasons why we might want to use many character states, one of which is hidden state models. So these are really important class of models that allow some hidden character to also affect diversification rates. And this is really important for avoiding false, spurious associations. Models of chromosome evolution, the states are the number of chromosomes, which can be quite large. And by geography, the number of areas, the number of possible states actually scales very rapidly with the number of individual areas that we're looking at. And when we analyze continuous traits, we'll discretize those into a large number of bins and then use discrete machinery. So in all of these cases, we end up with a model with a large number of states. So when we were writing TensorPhylo, we wanted to have a general model that unites the functionality that's available in a bunch of different programs. We also wanted it to be able to handle a large number of character states, and we wanted it to be fast. So a general state-dependent birth-death model includes lineage-specific events. So this is when an individual lineage speciates or goes extinct or is sampled or is treated. That's an epidemiological event where a lineage is sampled and removed from circulation. And all of these parameters can vary on the state according to the subscript as well as time. And we can represent for example, the speciation rate for a character with 4 states as a vector lambda 1 through 4. There are also tree-wide events. For every lineage-specific event, there's an equivalent tree-wide or mass event. So everybody is sampled or everybody is potentially exposed to mass extinction event, etc. The character state also evolves on the lineages. So there are anagenetic changes. Those are changes in state that are not associated with speciation events. And those happen with rate eta ij. And we can represent this as a matrix that's just like a Q matrix in a continuous time Markov chain. It's 2-dimensional, where the dimensions are the starting state and the destination state. There's also a cladogenetic state change process where a lineage in state I leaves one daughter in state J and the other daughter in state K. We can represent this as a 3-dimensional matrix, sometimes called a tensor, where one dimension represents the ancestor's state and the other 2 dimensions are the states for the 2 descendants. We also have a mass anagenesis where at a particular time, all lineages are potentially change character state. So a simulation under this model might look something like this. We'll start with one lineage in the blue state. We'll evolve it forward in time and it will have speciation events, anagenesis events, extinction events, and cladogenesis events. This is a treatment event, a sample that is also an extinction. There will be mass sampling events. Here, red lineages are more likely to be sampled than blue lineages. There will be standard sampling events that don't include extinction, like a fossilization event, mass treatment events, mass speciation events. All lineages speciate more or less simultaneously. Mass extinction events, lineages have some chance of going extinct, until we get to the present where there's a mass sampling event at time 0, the present. So all of these various modeling components are not available in all pieces of software. So this is a comparison of different pieces of software, including TensorPhylo, the default Rev-based character-dependent models, the Beast module BDMM, as well as 3 popular R packages. In terms of lineage-specific events, solid boxes indicate full functionality, so that's state and time dependence. Empty boxes mean no functionality, and partial boxes mean partial functionality. So in this case, these programs don't implement time dependence. In terms of mass events, programs generally involve mass sampling because there's species samples at the present, but may not always implement sampling over time, so mass sampling events in the past. And nobody yet has implemented mass speciation or mass extinction events. All programs involve anagenesis or implement anagenesis, may not be time dependent. Cladogenesis is not always available, and it's sometimes only partially implemented in terms of whether or not time dependent or whether or not both lineages can change. And mass anagenesis, which is currently only available in TensorPhylo. So when it comes to calculating probabilities under this framework, we have to solve differential equations that are of the form something that is linear with the total number of states. So this calculation grows at the same rate as the number of states. Then anagenetic changes induce n-squared calculations. So these grow, the number of calculations grows as the square of the number of states. And cladogenetic models include n-cubed operations. So as the number of states increases, the number of total calculations goes up by the cube of the number of states. So these are very expensive. But even models with anagenesis can scale slowly with the number of states. So to meet these challenges, we implemented TensorPhylo, which is a C++ library for calculating probabilities under this model. It doesn't do inference. It's meant to be plugged into programs that use it for inference. So we're using linear algebra library eigen for all of our matrix and tensor operations. And this is very nice because thinking back to our cladogenetic tensor, often there's a lot of zeros or other special structure in this matrix that eigen can recognize and exploit to really make the calculations much more efficient. We're also using adaptive numerical integrators from Boost and OpenMP for parallel processing. So I want to show you some performance comparisons. First, for multiple state models that don't include cladogenesis. So each of these dots is a program. The x-axis is number of states. The y-axis is seconds per calculations. Note that this is on the log 10 scale. Each dot is averaged over a range of tree sizes and parameter settings. So TensorPhylo starts off modestly faster than Rev-Bayes, but as the number of states increases, it scales very gently. Only diversity tree comes close. And again, doesn't implement most of the stuff that's available in TensorPhylo. So Castor in particular, I want to point out, is optimized for a large number of tips, not large number of states. So if we just look at 2-state models, as a function of the number of tips, by the time we have about 500 species, Castor is definitely doing better. So in the case where you have a large number of species, but not many states, Castor is the way to go. Otherwise, TensorPhylo is outperforming all other implementations. Now with cladogenesis, TensorPhylo is about, with only 16 states, TensorPhylo is about 100 times faster than Rev-Bayes for an equivalent model. That's with a single core, and if I use 4 cores, I get it like about a 2-fold speed up. Then as I increase the number of states, eventually it just takes so long for Rev-Bayes to calculate a likelihood that we don't even score it at like more than 100 seconds. But TensorPhylo scales much more gently, and the improvement of using multiple cores gets even better as the number of states increases. So these are fairly abstract comparisons of performance, just seconds per calculation. I want to show you an example of TensorPhylo in the wild. So with Carrie Tribble and others, I'm analyzing this data set of Carex. This is a genus of plants, sometimes called sedges, which has really interesting stuff going on in terms of chromosome evolution. So this tree has 742 species in it. And if you zoom in, you can see there's a lot of variation, even among closely related species, in number of chromosomes. There's interesting biology behind that, which I want to talk about. But there's between 5 and 66 chromosomes among these samples. And we're doing a chromohissy analysis. So in addition to modeling chromosome changes, we're also modeling hidden rate variation, which ultimately ends up in there being about 150 character states. And if we try to do this analysis in RevBase, it takes about 11,000 hours, otherwise known as 460 days, more than a year. If we plug TensorPhylo into RevBase, that comes down to 8 days, just a little over a week, pretty significant improvement. It's with a single core. If we use 4 cores, that actually comes down to 2.5 days. So an analysis that would have taken more than a year, now it can be run over the weekend. And there's some cool biology going on here for sure that I'm not touching on. You can go see Carrie's talk at Botany if you want to know more. So to wrap up, TensorPhylo, it's an open source library. It's publicly available on Bitbucket. But like I said, it's a library. It doesn't actually do inference. If you want to do inference, you're going to have to use RevBase, where it's currently available on a developmental branch. I'm also in the process of implementing an R version, which is basically fully functional. It's just not completely documented, but you can check it out if you're interested. And with that, I will take questions.